everybody to this week's edition of the Organismal seminar series. Before I introduce Nick Haddad, I want sort of a few announcements. First of all, don't forget wine and cheese after the seminar. I know none of you have forgotten, but just in case, it'd be good if that's your opportunity to talk to Nick if you have any questions that arise during the seminar. Followed by wine and cheese if you haven't had enough alcohol. We'll go up to Thompson House and have a beer or two with Nick before I whisk him off for dinner and more alcohol.
I'm ready.
He's already started. This is gin and tonic. Catherine wanted me to remind you that all these seminars are recorded and they're on the BGSA YouTube channel. So it's a great opportunity to go back, promote them, show off the kinds of speakers that we're getting in our seminar series. And we have a great example today of Nick Haddad, who is a William Reynolds Distinguished Professor in the Department of Biology at North Carolina State University. I think Nick's research, Nick is sort of synonymous with the idea of corridors and connectivity. He did a remarkable thing during his PhD with Ron Pulliam, so those of you who know source sink theory and big ideas in spatial ecology. He, during his PhD, set up one of the 6 long-term fragmentation experiments or corridor experiments at the Savannah River site in a longleaf pine forest. He set up these replicated landscapes of fragments from corridors, and broken corridors, set it up during his PhD, and then has since set this up as a long-term research site that's produced more than 60 papers and postdocs and PhD students, and is really sort of an icon, a poster child for this kind of research on the scientific basis of corridor science. And after his PhD, he went on to do a postdoc with Dave Tillman, Minnesota, where he branched into sort of more complexity thinking, the dynamics of food webs, and how different aspects of the physical environment affect diversity and stability in arthropod systems. And then he's today where he got his first job, which is quite rare in academia, and he's been there for 15 years. Along the way, he's doing lots of cool other things. He's a passionate butterfly ecologist. He's writing a book right now for the lay reader. I'm not going to give away what that's about. He's also a big promoter of corridors as a conservation strategy, so he has a blog/web page called Corridor Conservation. I encourage you to go see it. There are posts by postdocs and students and links to cool papers, and more importantly, I think a page that lists all the tools, sort of a GIS spatial model tools that resource managers, land planners can use and go to design a corridor, or and move the science of corridor science into application. So without further ado, by way of introduction, Nick, take the stage.
Thank you, Andy, and I want to thank you all for having me. It's been a real treat to be here. I've enjoyed meeting many of you so far, and look forward to meeting more afterwards. And it's just a thrill for me to be here and see your campus, interact with you all. I will start with a slight diversion from the main topic, but Andy set it up nicely, because I'll talk about what I would otherwise be talking about if it wasn't about corridors, just briefly with a few slides. And it does connect back to the main theme. But my other hobby and interest is to track down the world's rarest butterflies. And so this is a few that my lab has been looking into. And I want to tell you just very quickly a few of their stories and then how they connect back to the main theme. So this is the endangered St. Francis satyr butterfly, and if you could see it on the screen, maybe you can if you squint, you'll see me with a big smile on my face, because I'm in the middle of a swamp that is one of the homes to this very rare butterfly that lives in disturbed wetland habitats. Now if I step back and focus to its global range, you can see it here on this map in blue. You gotta squint to separate it from the speck of dust, but there it is, Fort Bragg, North Carolina. It's literally only found on a military base and an Army installation, and in that Army installation it is found almost entirely within bomb zones of that installation, where the bombs actually create the disturbance that keep the conditions right for the butterfly. Now going on to another butterfly, this is the crystal skipper, found on a stretch of North Carolina coast that's about 30 miles long, so it's just east of me. And this is a view, this is my former student, Allison Leidner, looking down its ideal habitat, which is coastal dunes. Now, before you accuse me of picking research sites so I can get to beautiful places next to the beach, I will tell you that this is the most excruciating place I have ever done field research, because here we're in North Carolina summers, so the sun's beating down with 95 degree F temperatures from above, it's bouncing off the sand up in my face from below. I'm running up and down the dunes to try to track down rare butterflies, and all the while I'm watching people on the beach enjoying the beach, which I cannot do. But this is the butterfly's ideal conditions. This is what most of its 30-mile range looks like. It's coastal development. This is beachfront property. The picture on the left is a town, Atlantic Beach, that's covered. The butterfly's habitat on the right are a bunch of little cottages along the coast. In that case, I don't know if you can make this out, but people have put their houses right on top of the dune. They wanted to be able to roll down off their deck to the beach and not give the butterfly or other wildlife places to eke out a life. And then my final rare butterfly, this takes me outside of North Carolina to South Florida, where the Miami blue butterfly lives. This graphic shows basically the former range of the Miami blue butterfly. It used to live throughout South Florida. It was considered a common, almost a weedy butterfly species. But now to find it, I get on a plane, head to Miami, then I get in a car from Miami, drive 4 hours to Key West, then get on a boat in Key West and go 10 to 15 miles out into the ocean to tiny islands. I mean, tiny, that are maybe fractions of a hectare in size, where this butterfly is still able to eke out its life. And the reason I show you these images to start out with is partly because I do love tracking down and understanding the conservation biology of the rare butterflies, but also because they do have a common theme that relates to my talk, and that is that all of these butterflies are rare, among the rarest in the world, very tiny numbers, because of loss of their habitat. And so where my talk really starts is thinking about the scale and scope of habitat loss. And so I start with this image of forest cover in the United States starting at about the time of European colonization, where, for example, the eastern United States is almost entirely forested. And if I had a pointer, I would focus in on the southeast region where I work, which is the area of longleaf pine savanna. And if you track through the next several centuries, at this resolution, you basically can't separate out the remaining area of longleaf pine savanna from the dust on my slide. Much of the habitat has been lost.
I've got a quick question about the butterflies.
Yes, go ahead.
Is this a situation where you have a splitter who has defined a bunch of different little populations and then each one might go extinct, but there's a bunch of closely related stuff that's nearby? Or are these really phylogenetically distinct groups that have nothing close to them that is abundant?
No, these are phylogenetically distinct groups. So just as an example, for the first one I showed you, which is the St. Francis satyr, it is a subspecies. There's one other subspecies that's found basically in a few fens in Michigan, but it's also endangered, but it's not nearby, and it also is endangered and very rare. And so, no, it's not the case. There's a broader discussion about that, but no is the answer.
Like if I, just wondering, if I split up, you know, something at a very fine scale, then you can have a bunch of little isolated populations
You could.
endangered, but that's clearly not the case here.
It's true. The second one I showed you, you notice it had no species name after the genera, but the species name, it's going to be defined as a new species. It's a paper's in review right now. Not by me, but by somebody else. Oh, thank you. Okay. So, but the real point of this talk goes beyond the problem of habitat loss, but to think about the problem of habitat fragmentation. So if we take a slide like this one, which shows oak woodland forests in central Wisconsin, much of the habitat is lost over the period of a century and a half as settlers moved in, cleared forests for agriculture. And the point I like to make from this slide is that yes, much of the habitat was lost, but it's not lost and the remaining forest is one large park in the center of the slide. It's left in tiny fragments that are really woodlots in this landscape, but this is consistent with what we find over the rest of the world. And so what I can show you today that actually links this slide, which I've shown for quite a while, to the rest of the world, is a new result that is coming out next week that Andy and I have been involved in with a group of other co-authors. This is a map of the world's forests. And it's one measure of the fragmentation of the world's forests. And so what it's showing is any color that's not gray is a forested area of the globe. And then it ranges from red, meaning very close to the edge of the forest, within 100 meters, to blue, which is more distant from the forest, greater than 1 kilometer. And what I first noticed from a map like this is that there are two places with lots of forest distant from the edge. They're like eyes shining out at me from the center of the map. It's in the Congo Basin and the Brazilian Amazon. And then just given where we are here, what struck me when I first saw this map is that the boreal forest is not a big blue area of forest. It is defined by, like the rest of the world forest, lots of edge habitat. And so just to summarize what we see when we look across the globe, we have this accumulation curve of the amount of forest that is at different distances from the edge. That's the histogram on the bottom, and then the accumulation curve above it. And what I see from this graphic is two things. First, 20% of the world's forests are within 100 meters of the edge. A short distance, they're close to the edge. And then 70% of the world forests are within 1 kilometer of the edge. Still close to the edge, still within a range that they can experience the effects of humans and their activities coming in from outside the forest. And just to put a finer point on this, focusing in on Brazilian forests, here I'm showing the changes from the original forest extent, and then I'll put up the current forest extent so you can get a sense for how this is changing over time. And so in the current forest, I use as bookends of the extent of fragmentation two forests in Brazil. At the one end is the Brazilian Amazon. The Brazilian Amazon has changed. Yes, there's some fragmentation, but there are still big areas of forest that remain that are distant from the forest edge. And so if you look at the histogram on the bottom left, the gray bars show the historic extent of fragmentation, that is distance to the forest edge. The black bars show the current extent. Yes, there has been change, but that change is nothing compared to a landscape that has been converted to for forestry and agriculture over hundreds of years, which is in the Brazilian Atlantic. There, most of the forest has been lost, and what forest remains is in tiny fragments that are close to the forest edge, that are well within the range of that they can be affected by people. And so I start by talking about the problem, habitat loss and fragmentation specifically. And where I want to pivot off to is thinking about what is a solution. And there, what I think about is the role of connecting landscapes via landscape corridors. And now I can put up lots of examples of where corridors are built in conservation to try to conserve biodiversity and ecosystem processes. And what I've done is started with a smaller example. This is one from my region in North Carolina. The dark green area on the top of this map is Duke Forest, so right next to Duke University, and then there's some county forest space that's down in the bottom right corner. The local land conservancy paid $100,000 to purchase this forest area, the light green, to connect via corridor those two forests. That's a small example, but the examples get bigger. This is an example of the Missing Linkages project from California. And it's an example of what I used to think of as just lines on a map, people drawing where corridors might go. But the Missing Linkages project, starting as a vision, has started to come to fruition in at least parts of it. And the one that I know for sure is the region, the corridors in the region between LA and San Diego, where 19 of these corridors have been purchased by the state at the price of tens of millions of dollars to conserve connections for mountain lions and other big animal and hopefully plants as well. And then what I think of as the grandfather of all corridors is the famous Yellowstone to Yukon corridor that runs from Yellowstone National Park, 1,800 miles up through the Yukon territories. And I like this image because it shows the glass is half empty, glass is half full view of corridors. The Yellowstone to Yukon project area is this entire light and dark green area. But the conserved area is the dark green area. And what you can see is that it takes up a good part, but still a fraction of the total space of the conservation area. And I can say that 3 years ago, the Nature Conservancy spent $500 million to conserve land to connect areas within this larger corridor. And so corridors have been popular, they're becoming more popular in conservation, and they'll become more popular still because not only are they being used to combat problems of habitat loss and fragmentation, but that in combination with other global changes like climate change. And so here I have a cartoon rendition of how species will change their range in response to a changing climate, moving to poleward or upward in elevation to get to cooler places. And so I have 3 examples from more mobile generalists to a dispersal limited specialist. And these species and their ranges in green are moving from one natural area to another, and only with the presence of connections through the landscape are they going to make it from one end of the range to the other as their ranges track climate. And one thing that we've learned through an analysis I did with Mead Crosby and colleagues is that current protected areas don't have the capacity to accommodate range shifts that are expected with climate change. What we did is looked at all protected areas, state and federal in the United States, and then made a histogram of their latitudinal extent. And what I learned from this analysis is that most reserves are small in latitudinal extent. And they're particularly small if you put that in context of the range shifts expected across plants and animals. That's what this red line is. It's from a review by Camille Parmesan in 2003. And it's the expected range shift given past evidence over the next 100 years. And it's on the order of 50 to 60 kilometers. And that review was done in 2003. A more recent review done in 2011 by Chen expects that range shift to be even further given projections for climate change. This is based on reviews across plants and animal species. And so given this momentum for corridors, it's then not surprising that there are big and bold plans to make bigger, more connected landscapes. I have one example from the state of Washington connectivity project. But there are many examples like this that cover big spatial areas. There are examples that cover small extents as well. This is a artist rendition of a wildlife overpass that would connect two areas of forest on either side of the interstate. And I love the artist's rendition of the plants and animals using the corridor, including, you'll have to squint to see this, but butterflies down in the bottom right corner. And so these corridors could, if working as envisioned, could be important and good things for conservation. But the question remains, do corridors work? I mean, that's what this bear is wondering as it's jumping off the road into a corridor. Should it use a corridor to get around? But more generally, do corridors work by serving as their superhighways they're intended for plants and animal species? And if they increase dispersal through the landscape, do they work by increasing the persistence of plant and animal populations? Does that increased dispersal have consequences for the diversity of species we find in otherwise fragmented landscapes? And that's the question that inspired my group to set up an experiment to understand the ecological role of corridors. And so what we did is we've been working in a forested landscape in South Carolina, and it's at a location called the Savannah River Site. And at the Savannah River Site, which is about 30 kilometers across, we set up 8 experimental sites shown by the orange dots. And what our experimental sites look like is, before they were created, is plantation pine forests, literally row crops of pine trees. And within those plantation pine forests, we created through harvesting networks of patches of habitat that are either connected via corridor. So there's one center patch about 1 hectare in size connected to another hectare-sized fragment. And then there's isolated patches that are the same size. Where we can test the role of corridors in affecting ecological processes. And just to clarify how these fragments were created, they're created by cutting pine trees in a plantation pine forest. And so what the corridors look like when they're first created is big open areas. This is ground zero. But from ground zero, communities of plants and animals quickly develop, and we've pushed along the process towards the natural ecosystem by managing towards pine savannas. And those pine savannas are maintained by very frequent fire about on the order of 1 to every 3 years. And so 1 to every 3 years, we work with the Forest Service to create controlled burns, 2007, 2009, and I had the treat for me to be out just at the point they were burning or just after burning this last week. And so this is what our plots look like nearly today. Here the remnants of a fire are still smoldering. And so very quickly, the ecological communities develop, and the point to emphasize is that our experiments create a contrast between the open savanna-like habitats that are productive and diverse and the surrounding pine plantation forest, where pines are planted at such a high density that there are not many plants or animals growing below the pine trees. And then, just to give you a sense for what these corridors look like, I have a 20-second video just to give you a ground level view so you can see more than what aerial images will show. And what these various images stitched together are showing are the kinds of efforts that we've undergone to test the ecological role of corridors, and some of these video images will come back in pictures as I go through the rest of the talk. And what I want to do is start by asking the question that we started with when we first set up the experiments. We're wondering, do these corridors serve as superhighways for plants and animals? Do they increase dispersal through the landscape? And I'm going to come back to giving you a broader overview, but I'll start with one species, coming back to my favorite group of organisms, butterflies, because that's how the project was started, literally around butterflies. And so with butterflies, we can catch them, many of them, mark them with barcodes on their wings, which you can see here in red, and then we can track marked individuals as they move through the landscape. And what we find is results like this one. Here I'm showing the proportion of individuals that were marked in one fragment that moved to another fragment that was either connected by a corridor or unconnected. And what we find for this butterfly, the common buckeye, is that twice as many butterflies move between connected fragments. That is, corridors do increase the rate of dispersal. Now we found this for common buckeye butterflies, but we found it for many other plants and animals as well. And so what I'm going to do on this graphic is summarize across many different studies where we've marked and recaptured plants or animals. And what I want to point out is that on the y-axis here, I've converted the units to essentially an effect size, a proportional increase in movement between fragments connected by corridors. So the point to make here is that an increase of 100% is doubling the rate of movement between connected fragments. And then if we look across all species, and I'll put up more data as I go along, the effect size is consistent and it's generally large across plants and animals. And it's true for butterflies and other insects. It's true for pollen, so we've tracked pollen movement between connected and unconnected patches for a variety of plant species. It's true of fruiting plants, so these are plants that are dispersed by birds, and it's true for small mammals. We've tracked through detailed mark recapture studies about 21 plants and animals, and for all but 2, we found that corridors increase movement. And so they do appear to be serving as these superhighways for lots of different organisms. Now, we did find 2 animals for which corridors didn't increase movement. This is the spicebush swallowtail butterfly and a cotton rat. These happen to be the 2 largest and most generalist animals within their taxa. And the story I like to tell here is that this spicebush swallowtail butterfly, we created fragments to study this butterfly. I collected thousands of eggs and caterpillars in the wild and reared them to adulthood so I could release individuals of known age into the fragments. We controlled the distribution of their food plants and nectar resources across hundreds of acres. And this was for a butterfly that didn't work, that didn't use that didn't use corridors. And so the point is that that was the start of the experiment. And yet it caused us that, what to me was a failure at the time, caused me to rethink when corridors might work and how to revision the study more broadly beyond one animal species. But generally, corridors seem to increase dispersal. And then we asked, does that increased dispersal have impacts that extend to broader consequences? And we have looked at whether corridors have an effect on increasing the diversity of species. My former student, Ellen Damshin, in the year 2000, set up these nested quadrats. This is actually the interior of several nested quadrats, and not just one of them in a fragment, but hundreds. I think she had maybe 15 of these sampling sites throughout our area. And it's hard to see on the resolution of the slide, but if you look closely, you can see by the big smile on her face that It was in her first year of graduate school. And yet she went on and kept studying the effects of corridors on plant diversity through the years. This is through 2010, but still to this day. We have plans to sample again in 2015.
She was in graduate school for 10 years?
No, no, no. She started in graduate school. She's still, she still sampled as a professor. Now she's actually has tenure even, so well beyond graduate school. And what, here's what she found. What I'm showing here is, again, a measure of effect size on the y-axis. This is a difference in the number of species between connected and unconnected patches. So here, a negative value would indicate less species in fragments connected by corridors. Positive values indicate more species. And here I'm showing data in the year 2000 when there was no difference. There wouldn't have been any difference because we had just created the experiments. There was no way for the plant species to respond to the treatments that we had imposed on the landscape. But then what happens over time is that the number of species in fragments connected by corridors increases relative to the number in unconnected fragments, to the point where by 2005, there are 20 more plant species in connected fragments. And just to give you a sense for the scale of this effect, on average, in one of our fragments, there's about 100 plant species. There's 20 more in connected fragments. That means there's 20% more plant species in connected areas. And this effect maintains over time. And actually, I could now extend this out to 2014. This effect seems to be maintained. Corridors have an effect on diversity. Okay, now I want to give you a little glimpse into what our next step is in understanding the role of corridors and some new analyses. I've showed you how corridors affect diversity across a variety of plants and animals, 20 species. And yet we're working in communities that have hundreds of plant species, potentially thousands of insect species. And